Hello and welcome to the inaugural webinar, Reimagining R&D by Vivpro. I'm glad you could join us today. We have an exciting topic around pediatric medications and availability of pediatric medications and how to accelerate that. Before we get on to our first webinar, I do have some housekeeping notes for you. To note that today's webinar is recorded. We are inviting you to have any comments or questions using the chat feature on your screen. If you experience any technical issues, please reach out to Holly at vipro.ai, and we'll appreciate your feedback by answering a brief poll at the end of this webinar. Having said that, it's my pleasure to welcome two excellent speakers and well-known researchers in the area and scientists in the area of pediatric medication. I will invite Dr. Lisa Bollinger and Allison Dunn to introduce themselves.
Hi, so thank you very much for having us, and thank you to all the attendees for your interest in the topic that's near and dear to all of us, pediatric drug development. I'm Lisa Bollinger, formerly Lisa Mathis. I'm a pediatrician by training, and after practice spent about 13 years at the Food and Drug Administration, where my last role was overseeing the pediatric and maternal health staff. In 2012, I went to industry to see drug development from the other side, and now have a more comprehensive understanding of pediatric drug development from both the regulator and innovator side of things. And I'm currently at Merck, where I'm the vice president for regulatory affairs, specifically for the general medicine therapeutic area. With that, I'll turn it over to you, Allison.
Allison, you're on mute.
I'm so sorry. Hi, everyone. Thank you for inviting me as well. My name's Allison. So I received a dual PharmD MS in pharmacometrics degree from the University of Maryland School of Pharmacy. I ended up enjoying my time so much that I pursued a postdoc with the Center of Translational Medicine, and now I'm currently a research assistant professor for the university. I've worked on a number of projects involving pediatric extrapolation and modeling. So this is a topic also near and dear to my heart. And yeah, I'm excited to talk about this with all of you.
Very good. Lisa, Allison, again, appreciate you joining here. I think our audience will be thrilled to hear from you. And I would like us to start off with the paper that both you and Lisa are co-authors. So maybe starting with Lisa. Lisa, can you share the strategic goals that you and your team have outlined to accelerate the availability of pediatric medication? And can you share that goal and a vision behind it?
You bet. So the goals really are to reduce the time between adult approval of a medication to the time that that product is labeled with pediatric data by approximately 50%. And then the idea of that really is to ensure that pediatric patients have access to evidence-based treatments. And because of the fact that we're in a period of refinement, which we'll talk a little bit more about later, we really are in a position to better use data to generate that evidence. And knowing what we know now, we believe that that evidence to support use in the pediatric population can be developed much more quickly than it had been in the past while we were learning how to do the studies and while we were learning how to use tools such as modeling. And then the second goal is really to renew pediatric labeling information for drugs that are older, that are off patent and don't necessarily fall under any of the pediatric legislation that incentivizes or requires studies in pediatrics.
Very good. So these are really important goals, and question always comes to mind, why? Why? So maybe Allison, from your viewpoint, why are these goals important?
Yeah, it's a good question. I think a big reason why these goals are so important is that ultimately it's going to help reduce off-label usage of medication in children, right? Evidence-based medicine is a standard we all live by, and it's important to know that the drugs we give pediatric patients at the dose we assume to be optimal are given with an evidence backing, right? Otherwise, we're violating this precedent that we set to be so important and to guarantee safety and efficacy. So by reducing the time to labeling, we're narrowing that window of opportunity for off-label usage first and foremost. But at the same time kind of goes for renewing pediatric labeling information for drugs without patent and/or exclusivity. So warfarin's like my favorite example in this. So warfarin is the most commonly prescribed oral anticoagulant in infants and children. And there's no actual pediatric labeling currently included. It is entirely used off-label in this population, and that's a crazy concept for such an old drug, right? But at the same time, it makes sense. Who's going to perform these clinical trials now? Who's responsible for that? This is where the use of real-world data and modeling really helps facilitate that. So this is something we're going to talk a lot about more as we go through the case studies as well. But I do hope it's clear up front, just from this like one example, that reducing time to labeling for new drug and increasing labeling for those off-use drugs is truly important.
Absolutely. And I think, before some of the laws we'll hear from Lisa were in play, it was even longer. So then the follow-on question to Lisa would be, are the current practices, laws supportive of what Allison talked about as the importance of reducing the time? Because warfarin is such an old drug, couldn't get through that, even still doesn't have pediatric labeling. So can you walk us through the history of what has the agency done, and you have had much role to play in that to promote pediatric drug development?
Sure. Thank you for the question. So there is a strong legislative history supporting pediatric drug development in both the United States and Europe. And a lot of that was, people were thinking about pediatric drug development very early. From, as you can see on the slide, even in the 1970s, people were thinking about the importance of having information and labeling, so that way pediatric patients could receive drugs that were determined to be safe and effective for them. But it was really in the late 1990s. And again, just a hat tip to visionaries like Murray Lumpkin and Diane Murphy, who were at the FDA at the time, that saw the criticality of getting data to support pediatric labeling. The FDAMA legislation added an incentive for pediatric study. And with that incentive program, with that piece of legislation, the floodgates were really open. And we started seeing a lot of data come into the agencies around using products in the pediatric population. And then with the passage of the Pediatric Research Equity Act and the Best Pharmaceuticals for Children Act in the United States, as well as the pediatric regulation in Europe in 2007, we suddenly saw this ecosystem developing where researchers and industry and government were working together to make sure that we were able to generate data to support the safe use of medicines in kids. Of course, then now, in today's day and age, we have substantial experience across government and academia and industry in doing those studies and recruiting patients and figuring out what the right type of data is that we need. But we also have experience in looking across the board at all the data that we have and using tools like modeling and simulation and extrapolation in order to generate the data that we need to label products for use in kids. Of course, too, the intent of the legislation in both the US and Europe is to get data on dosing, safety, and efficacy into the labels, so that way clinicians can make the determination about whether or not a product is right to treat the child that's sitting in front of them. One of the things that has been difficult is that the time between the time a product is approved in the adult population and the time that the data and evidence is generated and incorporated into labeling is so long that frequently clinicians have to use products off label in their patients, and they become so comfortable with them and there's access to them, and then it makes it really difficult to study those drugs because clinicians don't necessarily want to go into a study when they know that they can write a prescription for a product and treat a patient. And parents and patients might be reluctant to enter into a study with the additional blood draws and other requirements of a study if they can have access to the drug without entering into that study. So what ends up happening is that you end up in this loop where a product is approved, it starts being used off label, and that off label use incentivizes kind of this thinking of comfort with the product, and then it's more difficult to study. And so that's why trying to initiate studies earlier in the pediatric population will benefit everybody, because then people won't feel as though there's a downside to being engaged in the research. And there are some strategies in order to reduce those delays. One is the cooperation that's already happening between the US and Europe. They have monthly cluster meetings where they talk about the development of the programs. And this really helps in terms of synchronizing the requirements across regions. As we know, there are very few pediatric populations, and studies do have to be global in order to get enough patients enrolled in the study to have a meaningful outcome. So that the extensive collaboration that's happening across regulatory authorities is really helping to accelerate the time to get pediatric data. And then again, on the other hand, regulators are having realistic timelines for the conduct and submission of pediatric studies. As you know, Pravin, historically, there were times that companies would come in with very far out timelines for when they would complete the pediatric studies. And now the regulators are working very closely with industry to say, look, how long is it actually going to take you to get these studies? We're not talking about just setting a timeline of 10 to 15 to 20 years, but if it's only going to take you 4 years to complete the studies, let's use that as the deadline for submission of data. And then also talking about enrolling older patients simultaneously with the adult population. There's been a lot of work done in the pharmacometric space that really has demonstrated that exposure to medications in older pediatric patients, adolescents specifically, is similar enough to the adult population that there's no reason why in many circumstances those patients can't be studied simultaneously. Of course, we know there are some operational challenges with that. Those patients are generally seen at different centers than the adults. So that is a little bit harder for us to accomplish right now, but it's something that we can certainly work towards. I would say more importantly, being able to model and use extrapolation to leverage data from different populations and similar molecules. That's really what's going to help us accelerate moving those timelines sooner at this current time.
Very good. That's really an excellent background, and I think, Lisa, as you mentioned, it really allows us to think where we have come from compared to 1990s. As you said, it was difficult to do the studies. Early 2000, people were still trying to figure out how to work with the regulators, but a lot of blueprint has been ready on how to approach pediatric studies. In fact, there is a question which I will take after Allison covers the pediatric decision tree. So this is a good segue, including a question of what's extrapolation? What's the role of extrapolation? And maybe start with the full extrapolation, Allison, and once you're done with giving overview of the pediatric decision tree, I would like to ask a question that has come in the chat.
Sure, sounds good. Yep, so there are two different methods of extrapolation of efficacy, full and partial. This that I'm showing right here is actually a simplified form of FDA's pediatric decision tree. It's actually from an excellent paper that I've cited down here if you want to also take a look. But what this tool is it was provided by the FDA in their guidance on like general clinical pharmacology consideration for pediatrics. And it was intended, at least in the US, to help determine if a drug can actually undergo full extrapolation of efficacy, and that's option C right here. Partial extrapolation of efficacy is going to be option B, and then no extrapolation would be option A. And we're going to go through a couple examples of full extrapolation of efficacy. I think the best way to really understand what it is to kind of go through an example together. But if you just follow this decision tree, first thing it starts out for is, one, can you assume that children and adults have two things, a similar disease progression and a similar response to intervention? And if that's true, you can automatically either undergo full extrapolation of efficacy or partial. And the nuances I'll get into as we go along. But essentially, it's saving you from conducting extra studies in pediatrics. If it's full extrapolation, you just need one PK study to show that you have achieved similar drug levels. And then, of course, in every case, you need a safety study. There's no ever getting around that. And then in partial, you just have to do a additional PK/PD study to establish that exposure-response relationship. Does that make sense?
Very good. So let me take that question. I'll paraphrase and maybe bring in Lisa here. Lisa, the question basically asks, is efficacy not a priority in pediatric approval? Because, most are single-arm studies. Instead, is the priority safety? What would you like to share with our audience?
Yeah, I would say that while there are single-arm studies in pediatrics, there are oftentimes cases where we have to have a control arm, whether it's active or placebo, because we don't have the capability to fully extrapolate, as Allison just outlined. We may have a very different disease process in pediatrics versus adults, so you do need to establish that efficacy, or you may need a little bit more evidence in order to demonstrate that the pediatric patients are indeed going to respond to the therapy in the same way that adults are. But you're correct to say that there are some single-arm studies, and that the interest in pediatric studies, of course, also has to be safety. I will say that the European regulatory authorities have been much more open to the extrapolation of safety than the US health authorities have been historically. But the FDA has, in the recent years, made some statements about how we do actually extrapolate some safety from the adult population as well. But what we do know is that children have very different patterns of absorption, distribution, metabolism, and elimination. And so their exposure and response to the drug might be very different. And children are very different physiologically and anatomically as well. At a cellular level, they may have a different level of receptors for the drug. So their efficacy may not be as great or even meaningful, even though a drug has been efficacious in the adult population. The converse is true as well, right? That a drug could work in the pediatric population when it didn't work in the adult population. And then the other issue is safety. There are windows of vulnerability in the pediatric population because of the nature of the beast. Children are growing and they're also developing from a neurologic standpoint. So that does open them up to potential toxicities from a medication that should be characterized in the study of those drugs. So absolutely, the focus should be on safety. I think we're getting to the point where we are learning how to leverage the safety data from non-clinical and adult human studies into that pediatric population. And so certainly we do need to focus on safety, but there are also times when we're going to have randomized controlled trials where we will have to fully assess efficacy.
Thanks, Lisa. I think as we'll move into examples that Allison will cover, it will become clear that efficacy is still important. Mm-hmm. And I think, is it fair to say that the legislative standards to approve pediatric, even if efficacy study is not explicitly conducted, are no lower than what it would take to get an adult approval. It's just a different use of data or different use of science. Is that a fair statement?
Absolutely. Yeah, I couldn't agree with you more. The standards are the same. The difference is trying to think about what our prior knowledge is. What is the evidence that we already have on the table? And how do we fill the evidence gaps in order to preserve and protect the pediatric population and not enroll them in studies that are only going to reproduce data that we already have on hand. So this is more about being able to assess the existing data and how we use that existing data to get us to that evidence faster and more efficiently.
And the best way to learn that is with examples. So, Allison, looking forward to examples now.
I couldn't agree more. So we're going to start with full extrapolation of efficacy, talk about what it means, how it's previously been used, etc. So the very first case I want to talk about for full extrapolation has to do with partial onset seizures. Partial onset seizures are a type of epilepsy where onset is localized to one part of one cerebral hemisphere. They're also known as focal seizures for that reason. So why am I talking about this? Well, in pediatrics, focal seizures account for over 40%, or I think 50% actually, of all seizures in the pediatric population. So there's this like overwhelming and very clear need for pediatric labeling in anti-epileptic agents, right? Now, so what's the problem with this? Why is it so difficult to achieve labeling in pediatrics for focal seizures? And the problem historically was that it was thought previously that this type of epilepsy in pediatrics and adults was like vastly different, both in EEG patterns, etiology, response to medication, all these different things. This was the traditional thought, meaning that that very first question you see at the top of the pediatric tree, is it reasonable to assume disease progression and response intervention is similar? That was an immediate no. And therefore we couldn't even entertain the rest of the tree and the thought of extrapolation. So what ended up happening? Why did this change? Because I wouldn't be talking about it unless it changed. Well, what happened was a few different stakeholders saw that there was this unmet need that I discussed, and as a result, this project was conceived. And this was between the FDA, PEACE, which is the Pediatric Epilepsy Academic Consortium Extrapolation, and then the Center for Translational Medicine at the University of Maryland School of Pharmacy. And what they wanted to do was analyze all the various clinical trial data that had existed for commonly used anti-epileptic drugs to treat partial seizures and see if there actually was supporting evidence that contradicted this historical belief that they were different. And what ended up happening was they used PK/PD analyses, and they took all this real-world data and showed that patients aged 4 years and older actually had the same pathophysiology and response to treatment across like all these different agents that they looked at. And that's really great news. Right now we have some supporting evidence for full extrapolation. We can say that, yeah, in the future, this could certainly be used. But so what like actually changed as a result of this? Because of this collaboration, right, they're working with a regulatory body, the FDA. This kind of work was actually like compelling enough to elicit the FDA to release new guidance. Now, as a result, there's actually like official guidance document that states, yes, full extrapolation of efficacy from adults to pediatrics is in fact appropriate in the case of focal seizures, and we'll take it a step further, and this is how we recommend you do it based on all this prior research that we helped conduct. So now if you see how it's changed when we look at the pediatric tree, you can say, yes, response to treatment and disease progression is similar between children and adults. And furthermore, yes, there is similar exposure response when compared as proven by our PK/PD analysis of all this real world data. And what we're left with is that now, as I kind of talked previously, is option C. And in the case of future focal seizure treatments, the states that now we only need one PK study in children to achieve similar drug concentrations to what we're seeing in adults, because we're assuming we can extrapolate efficacy. If exposures are the same, we're saying efficacy is going to be similar. In addition to, of course, a safety trial. This is undoubtedly like a precedent-setting case, right? It elicited new guidance. And now we can have independent pediatric clinical efficacy trials and say that they're not actually necessary in the case of partial onset seizure. We've exposure matched essentially, and that's good. We'll go through another example that's similar to this, but you'll see why this is really important and how real-world data can be used to elicit and implement like actual change. But it really does, as you can see, rely on the effort and the dedication of all these different stakeholders to work together. And this kind of undertaking has to be collaborative, of course, in the case for these three stakeholders. This is actually kind of a big focus of the paper Lisa and I have worked on. And if there's like one major point you take away from this, I think it can be made now that we need the efforts of all these stakeholders to see changes like this to occur. So that is the first case for extrapolation of efficacy. I'm going to go over one other before we move on to the next one, and that has to do with schizophrenia. This is really a personal interest of mine because it's a fascinating disease. But schizophrenia, it's a psychiatric disorder that most often will present itself in like early adulthood. But early onset schizophrenia is possible. It's more uncommon. But as a result, when it does onset earlier, we often see it sporting this like much more severe symptom profile. And it's a huge burden, right? It's a disease has a huge burden at the individual, at the societal level. I think I included, the World Health Organization even ranked it as one of the most disabling and economically devastating diseases. So there is a big need to have good evidence-based treatment in pediatrics, even though it's less common. Now, so because early onset schizophrenia is so rare, there isn't a lot of supporting evidence to prove that those same antipsychotics used to treat adults were likewise as effective in pediatric patients before the project I'm about to talk about. But there is some good news, however, and that's that researchers weren't completely in the dark, right? There's both some evidence and some knowledge that we have to work with that I've kind of laid out here. And what we see is that early onset schizophrenia has been long thought to be phenotypically, neurobiologically consistent actually with adult onset. And we know that so much so because even the same diagnostic criteria has been used in both populations, and it's proven over time to be very accurate in diagnosing. Likewise, what we see in clinic is that the symptom presentation in pediatrics and adults are also like extremely similar. Although it's a more severe form in early onset, those symptoms that we see are almost identical. So in pediatrics, I believe it's auditory hallucinations make up a large proportion of the symptoms that we see. I think it's like 80%. And when they looked at like those auditory hallucinations in particular, they've shown to be have like very similar characteristics to those in adults. So we can start drawing all this like totality of evidence to see that, like they're really similar phenotypically and neurobiologically. And lastly, what we know is that there's this range of second generation antipsychotic agents that have been brought to market. And some had available electronic exposure response data for both adults and pediatrics or adolescents typically. Most of these trials used the PAN scoring system, which is the positive and negative syndrome scale, as their response of interest. And because it was so consistent, and that's typically what's used to diagnose, it makes kind of like this pooled analysis across all these different agents that we had, like very possible. And for all these reasons that you can see, it's very clear that extrapolation of efficacy from adults to pediatrics was clearly attainable. There's just one missing link, and that was actually identifying and like culminating all this evidence together and putting it in kind of in the right package. So how was that done? It's kind of similar to the first example, because as again, it was a collaborative project between the FDA and the University of Maryland School of Pharmacy, and they decided to approach this systematically again. So what they did was they used all that clinical trial data across a number of second generation antipsychotic agents, and they developed the very first quantitatively based framework. And they developed and validated this disease drug model and determined, based on all this clinical trial data that we have electronic records for, the model could actually adequately predict in the long term that the change in PAN score for adolescents, it was actually predictable based on that adult data. So basically what they did is they used some of that available data to build the model and predict that change in PANSS score in adolescents based on adults, and then validated and said, yes, it can accurately predict this in the case for other clinical trials. And so through this long modeling process, the team was able to determine that, in fact, this maximum drug effect for these different second generation antipsychotics were actually very similar between the two populations, and they proved this quantitatively. And this exposure response relationship also remained consistent. And yeah, it just was very ideal, right? They now have this like kind of indisputable, quantifiable evidence to our very first question, that decision tree, is response to treatment similar? And it's a yes. So I guess to summarize, with the availability of clinical trial data, work like this can continue to be conducted, and more models like this could be implemented to support like full extrapolation of efficacy.
Thanks, Allison. And I think, what the way I see it is that these are two really good examples of how to utilize data, and relates to the goal and vision you all put out in the paper. Because, and I personally know in schizophrenia, several sponsors started the trial, never could finish it because nobody would enroll in those trials. So having a goal to do an efficacy trial was almost futile, but needed efforts like these to bring in other knowledge to reduce the time it will take to pediatric drug development, and update the label. So, and I'll ask a question to Lisa, which is kind of continuation of the previous question. What's the priority as a parent, or even as a researchers? When I see something gets approved only based on PK, should I be worried, or should I be what should I be thinking about? Because seems like, the drugs are getting approved in pediatric just based on PK, which is kind of the other side of the previous question.
I think when you see that in labeling, people will say, the labels might say extrapolation was used and PK was done. I think it's important to realize that that's not the sole totality of data that was used to approve a product for the pediatric population. It was what was required as new evidence, so that PK data may help to establish the appropriate dose based on systemic exposure and everything else that's involved in pharmacokinetics. However, that data is coupled with existing efficacy data and existing safety data, or even other safety studies, as well as evidence that has been generated out in the medical literature and can be confirmed by the regulatory authorities. So even though the label may say that the approval was based on extrapolation with a PK study, that is only the new evidence or the new data that was required to generate the overall evidence to support that dosing, safety, and efficacy in the pediatric population. There is an abundance of data outside of that new data that's combined to provide that evidence.
Absolutely. And I think to Allison's point, the collaboration to bring that evidence to light is important. So we'll move on, Allison, to partial, the other part of the other side of the decision tree, to hear examples in the partial extrapolation area.
Sure. Yeah. So let's go over partial extrapolation. It should be pretty clear the difference once we start talking about it. And this next case, I actually want to talk about an example of just kind of outside the US in regards to treatment, because it is a really good example. And I know the FDA, EMA work very close together to talk about these things. But to do that, I'm going to talk about this case involving cystic fibrosis treatment. Cystic fibrosis, it's like the most common life-shortening autosomal recessive disease reported among Caucasians, at least. And it's caused by this mutation in the transmembrane conductance regulator, the CFTR protein, which results in this abnormal chloride transport. And the result of this is these thick secretions into vital organs such as the lungs, pancreas, liver, you name it, can be really devastating. And as diagnostic methods have improved for cystic fibrosis, what's ended up happening is that we can screen and diagnose infants well before even any symptom presentation happened. And that's huge, because if we have targeted therapies for these patients and it's caught earlier in life, we'll see a vast improvement in things like physical, psychological, economic, all these different burdens of cystic fibrosis because it is such a devastating disease. I think at this point, I've kind of driven the point home the need for treatments in pediatrics, right? Getting these drugs approved in patients faster means extrapolation of efficacy is a must. And there's this kind of clear chain of events that we've laid out, right? So what happened is the European Medicines Agency, EMA, had what I found to be one of the most compelling and detailed approaches to doing exactly this with this drug that I'm going to talk about, Symkevi, that treats cystic fibrosis. So in this scenario, the approval for Symkevi in children aged 6 to 11 years was granted based on partial extrapolation from adults and children aged 12 years and older. And this was considered appropriate because for a number of reasons, really. This was because there were similarities in genetics, similarities in molecular and physiological etiologies, all these different things that were noticed in cystic fibrosis between adults and children throughout basically the entirety of the disease course they found. And the steps of partial extrapolation in this guidance that the EMA released are extremely well described in their assessment report. I actually highly recommend people go look at this because it is quite impressive what they did, because the EMA essentially laid out exactly what you need to do for extrapolation. They indicated you need to prove safety and efficacy in adults, demonstrate that adult PK is predictive of pediatric PK, show that the drug has this predictive pharmacodynamic effect, provide evidence of efficacy, evaluate safety. They give like a very detailed approach, which can be really beneficial for sponsors and people who want to bring more drugs to market because they have like a roadmap to do that. And basically they gave, again, this incredibly well-defined point A to point B network. So in this case, there are two things. There's the sweat chloride test, was selected as this pharmacodynamic biomarker because it demonstrated how chloride levels improved equally when they looked at adults and pediatrics. It was equal. Likewise, they looked at the lung clearance index, which was utilized to prove that treatment was being efficacious in adults and pediatrics. And what this means is that now we have these very well-defined PD biomarkers that can be reliably used. And they proved that through all this work. And going back to the FDA and this pediatric decision tree, this would translate now into option B, right? Partial extrapolation of efficacy. Now, unlike in the case of full extrapolation that I discussed previously, where we only need one PK trial, now all that we need in addition to that is this PK/PD study to establish that exposure response relationship with these PD biomarkers that we indicated are well qualified and good to actually use in these studies. So all in all, I think that this really should be considered like a hallmark example, at least, and this is my personal opinion, of how these physiological biomarkers are used to draw these comparisons in efficacy for pediatric patients, while also using what is a clinical endpoint to prove efficacy. It's really eloquent what they did. I thought this was a great example. I do have one other example focusing back to the FDA just so we can get kind of a more holistic view. And this has to do with cardiac arrhythmias. So what I went over before was an example of how partial extrapolation was conducted at the EMA. Here in the US, a really good example is sotalol. And sotalol is a class 3 antiarrhythmic agent. It was originally approved by the FDA for like life-threatening ventricular arrhythmias. Now, however, its use has been expanded. It's now also used to treat patients with AFib, to maintain sinus rhythm, and a few other things. So conducting efficacy studies for antiarrhythmics in adults already, just looking at adults, is extremely challenging for a bunch of different reasons. One is that the endpoints can be very uncertain and they have really high variability. And subsequently, because of this, you need much bigger sample sizes basically for these studies. For something that's already like so challenging in adults where our patient population is larger, and all these other things, you can see how trying to have true efficacy studies in pediatrics is even more difficult. Large sample sizes aren't practical in the case. There's ethical issues as well with having these very large sample sizes. There's a lack of interest from patients or from parents that Lisa kind of went over, wanting to actually enroll their child in the study. And also, like I said, just the patient pool is so much smaller just by nature. So what we need to do is find a way to circumvent this, right, for these efficacy trials. This is a very clear problem. And if we want to get drugs approved in pediatrics like sotalol, we have to find a solution to this. So in the case of sotalol, it was demonstrated that to achieve pediatric labeling, one PK trial and another biomarker trial were all that was needed for at least for patients aged, I think, like 3 days to 12 years. So how'd they do this? The sotalol biomarker trial assessed the antiarrhythmic and beta blocking effects on QTC and heart rate, and they did this with an ascending titration regimen. And what happened was their analysis showed that class 3 pharmacologic effects in pediatrics were very consistent with what they observed in adults. And for that reason, partial extrapolation of efficacy was actually deemed appropriate and subsequently implemented. As a result of this biomarker study, pediatric dose recommendations were added. Basically, what were they able to do is make this case that why were they actually able to achieve partial extrapolation of efficacy, even though there were all these problems that I kind of previously described. So there's 2 primary reasons. First, it's that it was very clear that you could have this reasonable disease progression and response to treatment that was assumed to be the same between the 2. There was no evidence to suggest that there were differences in pathophysiology or biology. And they're not known to be differences that exist actually in regards to etiology. So prolonging the phase 3 repolarization reduces ventricular rate, and potassium channels are the same in these 2 populations, right? That's kind of something Lisa said. In some instances, it's not always the case. The actual like site of action, it could be different between the 2, but in this case, it's not, and they were able to prove that. And the second reason was that just why this was actually achievable was because of how reliable those biomarkers selected were, QTC and heart rate. They were inarguably the best metric in the case of antiarrhythmias, and therefore could like undoubtedly predict efficacy. So in small children, obtaining QTC and heart rate is very easy to do, right? It doesn't depend on some things that maybe a younger generation wouldn't be able to perform. So things that maybe require like linguistic ability, advanced cognitive ability, right? It doesn't rely on those things. So you can make a really good comparison between pediatrics and adults. And what I think we should learn, at least from this example, is that treatments with biomarkers that are so generalizable, we should automatically assume and assess clinical efficacy in both populations and see that they're probably ideal candidates for extrapolation of efficacy. And they should be pursued as such, basically, because they are just so similar and they're not reliant on these kind of age reliant differences, if that makes sense. That's, I think, all I have for, yes, that's all I have for these examples. So thank you for letting us talk about this paper. This is something Lisa and I really enjoyed working on. And so it is very fun getting to go into a deeper dive of all of our different examples. There's a QR code I think we included. If you do want to read the remainder of this paper, there are a couple other examples that I just didn't include for time reasons that I think would also be good to take a look at.
Very good. No, these are excellent examples because I think if you look at both cystic fibrosis and the cardiac arrhythmia, especially cardiac arrhythmia, in adults, one need to prove using mortality morbidity benefit. So having a pathway for pediatric approval based on biomarker, it's an amazing step forward. So in fact, there are a couple of questions, actually several questions in the chat, but let me take one question that came up. And I believe it may be about the last case study. Did they have to prove the biomarker was prognostic in the pediatric population besides predictive as well?
That's a good question. I would assume so, but I'm honestly not entirely sure, um, just offhand. That would make sense, right? But yeah, I'm honestly actually not sure what like that if it was the methodology that they did for that.
Yeah, I think as a part of, and Lisa, if you have comments, feel free to add. But as a part of the
Mhm.
first deliberation, is the disease similar? The biomarker is part of the story. Although I think the question gets to, did they have to prove prognostic? This is more handled in my experience using more of a clinical knowledge. There is no data. I mean, there are a few instances where prognostic biomarker has been proved using data, but there's really, I think, a lot of clinical knowledge, and including, I'm not sure if it was done for cardiac arrhythmia, but FDA uses advisory committees for this kind of question. So, Lisa, any comments on that? Whether it's important to prove whether the biomarker is prognostic?
Yeah, it's critical to prove that, right? Because if you're going to be relying on it, you have to be able to rely on it. Um, but but I think the point is that you don't always have to generate new data. That data may already exist to support the reliability of a biomarker in order to support extrapolation. So again, that's the work of the drug developer to really dig into the existing data and to try and get to that primary data to ensure that it supports extrapolation. And if it exists or if you can generate it, then obviously you may use it. But word of mouth or hearsay is not sufficient for people to use that biomarker as a linkage to other data. There has to be evidence to support that.
Very good. Two questions that have come up, and either of you can take this. Very interesting, actually. We have been talking about extrapolation so far, but the first question is: can you speak about the feasibility and challenges of developing novel drugs specifically for pediatric indication without an adult program? So all of our examples have been something approved in adult. How do I get to pediatric faster? But what if there is no adult program?
Well, I'd be happy to speak to this. And it's funny because as I was thinking through my list of challenges, there are really challenges that haven't changed for the 25 years that I've been doing this. I think that the first one is thinking about what is the appropriate subpopulation of the pediatric population that's affected by the disease or condition and needs treatment. So really defining who is that population that you need to study. And then I think another challenge is developing appropriate endpoints. Oftentimes, we don't know what those endpoints should be, or because of the limitations of the pediatric population, they aren't mobile, they're not walking before a year of age, and they're not talking before 2 years of age. So you have to be very careful about how you measure the endpoints. And I mean, I think Pravin can remember the struggles that we had at the FDA trying to figure out if we're developing a medicine to treat preemies, neonates, with blood pressure medications, how do we know what the target blood pressure medication is for an infant that's been born preterm? Because that's not a normal physiologic state. So getting that endpoint right is really important as well. Obviously, figuring out if you're going to have a comparator arm and what you're going to include in that comparator arm. So you can have placebo if there's clinical equipoise, if we don't truly know the benefit risk profile of a product in the pediatric population. Or if there are other products approved, then ethically you can't put a child who needs treatment on placebo. You have to use that active comparator. But we also have situations where we have standard of care that's not approved, but is used typically. So how do you use a standard of care when you don't have a quantitative treatment effect as a comparator? So that's always a real challenge to think about how you're going to design your study. And then obviously enrolling enough patients. Pediatric patients make up about a third of the world population. And even if you say half of those children are sick and need treatment at some point in their childhood, it's a plethora of different diseases, right? So that subpopulation that is both eligible and available for study is small. So then we have to leverage a lot of clinical sites and go across regions, which speaks to the importance of different regulatory authorities across regions collaborating as they do. I think one of the things that's become very obvious to me in the last 25 years, and I'll stand by this as well, is that dosing becomes really important in the pediatric population. You can get everything wrong or everything right and just have the dose wrong, and you may end up not being able to demonstrate that tied efficacy or efficacy on its own. And then again, the other issue is the formulation. Younger pediatric patients can't take an oral dosage form, a solid oral dosage form, and developing pediatric formulations is a lot harder than anybody ever thinks. So I always encourage companies to start working on their formulations long before they even think that they need to be doing that, because that's always a big challenge. So that way you can deliver the right dose. I'll stop there in case Allison or Profit have anything else to add.
Allison?
Summed it up almost entirely. It was perfect. I don't have much else to add. I think, yeah, just the nature of having pediatric clinical trials is going to be difficult, and that's going to be a big limiting factor. I don't know if you have anything else to add, Robin.
Yeah, no, I think both of you summarized. What I would say is, and goes to the points both of you mentioned, to the question that was asked. If it's a purely pediatric program and there is no adult program, it's making a case for that disease, and perhaps even looking into epidemiologic evidence, any observational studies that are available. What I would say again, none of us are here at work at FDA, but at least in our experience, FDA is very open to getting educated on that science, and it's on incumbent upon the company. That's where public-private partnership plays a role to make that case for that particular disease and population. How can this be more efficient way of doing? Because the alternate always is go do a large randomized clinical trial and see you after a few years. And we have been there, done that. Except if it's a very prevalent disease in pediatric, those trials don't happen. So FDA is very open to that.
Yeah, and I think that all of the issues that we just discussed are really amplified in that neonatal population. I know somebody asked about neonates. All of those problems become much more difficult. And the one piece of advice I would give is always make sure that you have pediatric clinicians engaged in helping to design the studies and figure out what the true problem is that you're trying to address is, and then also to work very closely with the regulators. They have a lot of experience and can provide a lot of guidance around how to develop a product in the youngest pediatric population.
Perfect. A lot of great questions are coming up, so let me kind of follow through those questions. We covered the pediatric. I think a quick follow-on to that is a more question on the preclinical studies. Should there be more of emphasis on preclinical studies on more physiologically relevant animal models such as bovine, porcine versus rodent? Any thoughts on that, using preclinical evidence as a part of your data package?
I think from my perspective, again, it depends. You can tell I was a regulator for a while, right? So it really depends on the disease and what you've seen in the non-clinical studies. Obviously, you need to have sufficient information on what you need to look for in terms of safety from those preclinical studies. So sometimes actually using models other than mice or rodents is critical. Sometimes it's not. And we also have to be careful about how much we require preclinically, only because that's not always the answer to do more preclinical studies. I think too that we have to think about not only what species we're using, but what are the ages of those species and how is that relevant to the pediatric population. But absolutely a robust contemplation about the preclinical data is critical. And that includes considering both the species that you're looking at, as well as the age of those animals that you're studying.
Very good. And I think the totality of evidence is the name of the game there, is to really make
Yes,
that
thank
totality
you. Absolutely.
argument. An interesting question came up on these webinars. We don't usually or don't want to talk about RIA, but the question came up, and I'll direct this to Allison. Allison, can you comment on how RIA was leveraged and RIA stands for R&D Intelligence Assistant, which is one of the Vivpro product, was leveraged for these case studies, or can we do this for similar future cases?
Yeah, I think it's honestly a good question. I was hoping we'd get to talk about it because I did really think it was such a good tool to get to use. This isn't them paying me to say this, I promise. Like it is a genuine just opinion of mine.
No marketing, absolutely.
Yeah, for sure. So it was used. What we did when we were diving into this in the beginning, like we knew just because everyone here has a lot of experience, but we wanted to see like what was the actual change over time for pediatric approval from when adult approval had occurred. And RIA made it like super easy to access all this different labeling information, and to identify when extrapolation of efficacy or partial extrapolation of efficacy actually had occurred. And I think it probably reduced our time to conduct all this research, I would say, from like what maybe would have taken 6 months into I think we did into like a couple of weeks. So it really is like as someone who wanted to do this very efficiently and get this paper out here, it was a really great resource in that way. In the future, I think it can be used similarly, just actually being able to collect all this real world data and digitize it. It sounds like straightforward, but it is not simple. It takes a lot of effort. It takes a lot of, again, collaboration, diligence to want to do this and seeing a need. And so having a tool where you can make that a little bit easier is a huge benefit.
Yeah, I kind of equate it to archaeology. We can now use lasers to find ancient civilizations in the Amazon versus trying to do it a shovelful at a time. And in the pediatric world and in drug development in general, we're sitting on a mountain of data, and having tools that allow us to access that data more efficiently and more completely really helps us to be more informed as we go into drug development. So I agree with you, Allison.
Absolutely. And, I think I've seen with a few other examples, including publications, people have found a precedent and then have pretty much followed the blueprint. And I like the archaeology reference, Lisa, because that's kind of, at the end, these are needle in a haystack kind of problems. Once you find them, then it becomes a blueprint of what you want to do. Or even knowing that nothing exists in the area, it preps companies to make the case on their own. So both sides of finding something is useful, but also confirming that something is not there has been quite useful in our experience. So with that, I would like to thank Allison, Lisa. Great discussion. Again, I wish we can continue and look at the remaining case studies that you have. I invite everyone to look at their paper because it's an excellent summary of what has happened and some of the precedent setting case studies. Hopefully you will join us at the next seminar at a webinar that will be how FDA and payers utilize RWE, similarities and differences. That would be by Dr. Brigham Hyde at Eversana. It will be in August. We will send you a follow-on announcement on the date as well as a refined title. We'll add you to the mailing list, but if you are if you're willing, follow us on LinkedIn. The QR code is right there. And what we'll do at this minute is, in last 1 minute, we'll flash a poll for you to give us a feedback, and it's a simple NPS response poll. Would you recommend our seminar to your colleagues? So go to your polls section of the webinar link and let us know, because this is what we want to do. These are not uh, commercial webinars. We really want to engage discussion. We want to invite researchers like scientists like Lisa, Allison, to conversations, so we as a community know where the science of drug development needs to go to reimagine the R&D. With that, thank you very much, and see you in August.
Thank you so much.
Yeah,
Bye bye.
thank you, everyone. Yeah.